Now, when we look at lipids and their structure, we can talk about the differences in their solubility and melting point just based on some of their chemical properties. And I kind of hinted at this before, but as you might imagine, because these are hydrophobic and they are long hydrocarbon chains, as you change that chain length, you're going to change their solubility. So these fatty acids are going to be less soluble in water the longer that hydrocarbon tail. And again, the more hydrocarbons you put on there, the less polar the molecule, the less soluble in water. Their melting point also is going to change depending on that chain length. So again, melting point will decrease as the chain length decreases. Again, this is going to be based on their ability to stack on each other and their interactions. So let's think about this. If you have a very, very, very long fatty acid, this actually stacks really well with itself. And this is because of van der Waals. We talked about this at the beginning of the semester. So just remembering our types of intermolecular forces. When you have these very, very long molecules with lots of surface area that are hydrophobic, they actually interact with each other. They pack with each other very well. And because of this, it makes them a little bit harder to pull apart. Remember, when we're melting, we're pulling them apart. So the longer they become, the higher the melting point, and the shorter they are, the lower the melting point. Now, the number of double bonds also is going to alter that melting point. And remember, that's because of this conformation that we see. So the more double bonds there are, the more kinks there are in the molecule, the less better they stack. So again, saturated, solid, unsaturated, or double bonds, they tend to be more liquid. That means their melting point is lower, right? They've already melted at room temperature. They're a liquid. So again, to remind you what that looks like, the saturated have these nice linear extended conformations that stack really well. The more double bonds you put in, the more kinks you form. And that's again because we in nature only find cis conformations of double bonds. So remember the difference between a cis double bond and a trans double bond. In nature, these are the only types of double bonds we found naturally made in fatty acids, which again kink the chain because of their this cis conformation, making them stack less and making them have a lower melting point. So these tend to be liquids at room temperature. So again, saturated fatty acids are going to pack pretty orderly, and they have very favorable interactions with each other. Unsaturated or cis fatty acids pack less orderly, as you can see in the image below, because of their kinks, and they have less favorable interactions with each other. They can't stack very well. So it takes less energy to disrupt something like this because they're already disordered, they're already disrupted in a sense. It takes more energy to disrupt or pull apart these because they have better stacking.